Hi everyone, this is the last of the mini lectures for Chapter 13. In this lecture, we will be talking about B cell response. When we talk about B cells and their response, we are talking about the activation of B lymphocytes. And that activation will cause clonal expansion and antibody production. And in order for these to happen, for a B cell to be activated, it needs to be activated by an antigen, just as the T cells do. So if we look at this table, which is table 13.6, we have the different steps and players in B cell activation. We have a microbe coming in where we have a B cell. It will bind to a pre-committed receptor to that particular antigen. That antigen then, or microbe, will be processed within, phagocytized and processed by the B cell, and then presented on its MHC2 receptor. This will allow it then to do communication with a T helper cell, which, as we just learned, we know there's a lot that goes on when this T cell is activated. In addition, once it has done this cooperation and recognition with the T cell, we have interleukins that are going to activate this B cell. So down here we have the activated B cell, and this activated B cell then is going to differentiate into three types of cells. It will do regulatory B cells, which will secrete IL-10 and will regulate the T cell response. It will also then make more memory B cells with this particular antigen. And this allows these cells to be in circulation looking for the exact same microbe to come in. And then the third type of cell that will be produced once B cells have been activated are plasma cells. And plasma cells are very short-lived, and they make antibodies with that certain recognition of the microbe from the very start. And they will put out a bunch of these antibodies into the lymphatic circulatory system, and it will be looking to attach itself to that same antigen. So hopefully this gives you a nice brief overview. We'll probably do a little bit more of this going forward so you have a little bit more idea. But this is a great table to look at. So how does the antigen interact with the antibody on the B cell? And what happens is we have two different parts to the region that will bind the antibody, the antigen. We have the hypervariable region of the antibody, and this is the site on the antibody where the epitope from the antigen will be bound. And the amino acid content of this region is extremely varied, which makes sense when we realize that each of these antibodies are specific for one antigen. So that specificity of the antigen binding sites for the antigens is similar to enzymes and substrates. It's very specific, and it's for that one particular epitope. So this is another table that will be helpful in looking at the functions of antibodies. Antibodies, once again, are these Y-shaped molecules here. And what we see in the first picture is it coating the surface of a bacteria. And what this does is it'll often prevent it from doing normal functions and reproduction. In addition, these antibodies can stimulate something called opsonization. And this is a process that makes the microbes more easily recognized by the phagocytes. So it actually becomes like a handle on these bacteria that allow the phagocytes to grab on and then phagocytize that bacteria. In addition, it can also do neutralization. And in neutralization, the antibodies will cover the surface receptors of a virus or an active site of a microbial enzyme to prevent it from functioning normally. So these are all kind of the ways that it does this, with that bacteria up here, the bacteria here to be phagocytized, and then here we're looking at a virus. And then also these antibodies have the ability to be binding more than one bacteria at a time. They have 2 binding sites. Each end of that Y has a binding site, and this allows them to actually aggregate or pull together a bunch of bacterial cells. And this often makes those microbes immobile and enhances their phagocytosis. There's also interaction of the antibody with complement. And in this sense, it plays a role in getting that hole in the bacteria that will end up lysing the bacterium. And then lastly, it can play a role by binding an antitoxin and neutralizing bacteria's antitoxins. Okay, so that's kind of the summary of antibody functions, and it's mainly dealing with that variable region because that's the part that's specific for a particular microbe. But what about the other end of our antibodies? And that would be the bottom part of the Y of the antibody. This is called the Fc fragment, and it actually has important functions too. It contains an effector portion that can bind to receptors on the membranes of cells, especially cell types like macrophages. When it does this, the binding is dependent on the cell's role. So opsonization is often because of that attachment of the antibody to foreign cells. Will then bring those foreign cells, and the Fc portion will bind to the phagocyte. So actually grab onto the phagocyte to allow phagocytosis. In addition, on an IgE, the Fc end, that constant region of the antibody, will bind to basophils and mast cells, which cause the release of allergic mediators. So as we're looking forward, there are different classes of the immunoglobulins, and they're mainly based on isotopes. So they're structural and functional classes of immunoglobulins. And the one that we are going to spend the most time on is IgA. IgA is very important because it coats the surface of our mucous membranes. And it also is suspended in many of our liquid or mucus areas. So saliva in the mouth, tears in the eyes, mucus. We have also colostrum, which is the earliest secretion of breast milk. And there's a lot of IgA in the colostrum, and this will coat the gastrointestinal tract of a nursing infant. So already we're getting some of that specific immunity transferred to an infant. And they also provide specific immunity against enteric, respiratory, and genitourinary pathogens. So we have a table here. This is table 13.8, and this shows the characteristics of the different immunoglobulin classes. The one that we talked about was the IgA, both as a monomer and then here as a dimer, which is the 2 of them bound together here. You can see some different things, the number of binding sites, but the big one here would be looking at the biological function, seeing what these function for. For example, IgE has something often to do with allergy, and here IgD, it is a receptor on B cells that trigger that molecule for B cell activation. And you can see each of these has a particular function. So mainly knowing the different immunoglobulin classes and their function. And then lastly, I want to talk about that antibody production over time and what it looks like between that primary response, so the first time that you've been exposed to a particular antigen, and then that secondary response when you are then challenged again by the same antigen. So in the yellow here, this is table 13.9, this yellow-orange box, this is showing the first or primary response. So we have a first encounter here, a first exposure to an antigen. There's a latent period. It takes a while for these cells to get up and running, recognize it as foreign, start to proliferate. And you can see we have IgA is the red, the light blue is IgG, and then the total of those 2 is the blue. And you see we get this nice response, but it takes a little while. And then after a variable time interval, this can be a short amount of time or perhaps a long amount of time, but our body should always be prepped for a second response. We get second exposure to that original antigen. And what you see here is there is no latent period. It is immediately recognized as foreign, and there is an immediate proliferation of our IgA and our IgG. It's a much higher peak, and it is a much quicker response. So this should tell you why it's important that we have a first exposure and then that memory and specificity of each of our receptors. And that's the last of chapter 13.